I'm so excited to be here. Biomarkers and outcome measures are my favorite subject. Um, my little sort of claim to fame is that I went back to school to get a PhD when I was an old lady in order to cure my kid. And by the time I finished my PhD, it turns out what was really needed were biomarkers and outcome measures, not any more compounds. So I've been working on biomarkers and outcome measures ever since. And I'm super excited to have this illustrious group here with me today. I'm going to start with Pat at the end with the amazing green glasses. Um, I had the pleasure of being on a committee recently with Pat for almost a year, and it was really a fantastic experience. That's how I got to know her. So Pat Furlong is the founding president and CEO of Parent Project Muscular Dystrophy. It's the largest nonprofit organization in the US solely focused on Duchenne muscular dystrophy. Duchenne, if you don't know, is the most common fatal genetic childhood disorder, which it affects about 1 in every 5,000 boys each year worldwide. It currently has no cure, but for anyone who's been following the news, you will know that there are now 7 approved treatments for Duchenne specifically, including an enzyme inhibitor, a specialized steroid, antisense oligonucleotides, and gene therapy. It's really been a huge groundbreaker for rare diseases. So I'm going to tell you a little bit more about Pat, just so you really know about her. Um, she entered the field when doctors diagnosed 2 of her 4 children, her 2 sons, with Duchenne, her sons Christopher and Patrick. She didn't just accept, no for an answer. She immersed herself in Duchenne. She worked really hard to understand the pathology of the disorder. She learned all about research investment and how to take care optimally of children with Duchenne. Her sons did lose their battle to Duchenne in their teenage years, but she keeps working in their honor and for everybody else affected with Duchenne. So that's Pat, and she's a real hero. And so I guess I'll keep introducing. Okay. So I'm going to go on next to Dr. Matthew Ellenwood. That's this one right here. Um, he is a veterinarian, scientist, researcher, and very passionate about MPS. Um, he has been serving for more than 4 years as the chief science officer of the MPS Society, the National MPS Society. He's also a comparative medical geneticist. Should I have said that instead of veterinarian? They're both. Okay. He's got over 2 decades of research experience with mucopolysaccharidosis and mucolipidosis. Aren't you proud of me for leaving those in full like that? Type 2 and 3. He's a professor emeritus at Iowa State University, where he conducted both basic and applied research on the neuropathic MPSs. He has experience in just about every type of gene therapy delivery, and including intraventricular enzyme therapy, systemic IV, AAV9-based gene therapy, intraparenchymal, CNS-directed, etc. However, when he entered the field in 1998, there were no treatment options beyond stem cell transplant for MPS. Now, MPS has 5 drug approvals, national implementation of newborn screening for 2 MPSs, state screening for a 3rd MPS disorder, and potential accelerated approval on the horizon for both gene therapy and 2nd-generation ERT. It's another amazing story for the rare disease community. Okay, one more to go. Dr. Celia Goeldner. Goeldner? How do you say your name?
Goeldner.
Goeldner. Dr. Celia Goeldner is a neuroscientist and a clinical expert team leader and clinical scientist at Hoffman La Roche. She has been at La Roche for 15 years. She started off as a researcher and she's worked her way all the way through to the early clinical development field. She has been working to translate drug candidates into medicine for patients with psychiatric, neurodevelopmental, and rare neurogenetic disorders. Most recently, and this is how I've been able to meet her, she's been working on drug development for rare genetic disorders of the 15th chromosome. So I just am so excited to have all of you three here, and I just want, they're pioneers, and I just want you all to just give them a hand. So let me start first. I'm going to actually get you to do some definitions. So this is because we really need to define what we're going to talk about today, which is biomarkers, outcome measures, and surrogate endpoints. So would one of you volunteer to define what is a biomarker? There's a slide right there.
I'm happy to talk about biomarkers. Biomarker can be anything you measure about a physiological system. It could be imaging, X-ray, MRI. It could be something you measure in a tissue or something you measure in a fluid, CSF, urine, plasma, etc., that in some way is associated with the disease pathology you're trying to treat. There are all sorts of biomarkers. There is a very small group of what we call biomarkers that are validated by the FDA. That is in a specific kind of very arduous and very painful criteria. We're not talking for the most part about biomarkers for rare diseases being validated. We just don't have the wherewithal to make that very arduous lift. But we would like reviewers to accept biomarkers as a valid way to measure aspects of disease related to clinical endpoints, and that is a good handoff to the next topic, I think.
Okay, that leads us to the next definition. What is an outcome measure? Would either of you like to take this one?
Oh, I'll take it. So an outcome measure is a measurement of clinical function, or can be patient reported or clinician observed, and it reports on the patient's health status and disease advancement. And it's in certain fields, it's quite difficult, especially in CNS brain disorders, to have those outcome measures that are objective, that actually directly measure change. They're often reported, which makes drug development more complex.
Okay, thank you very much. So biomarker is objective, outcome is a little bit more subjective, although clinical outcome measures are measured by doctors. Okay, what is a surrogate endpoint?
Sure. A surrogate endpoint is an endpoint that is a surrogate or describes something that's reasonably likely to predict benefit. So a surrogate in a rare disease, for instance, if there's a protein missing and you can measure that protein, and that protein is expressed rather than not expressed based on the genetics, then that protein could be a surrogate marker for benefit in that particular disease, because it's reasonably likely to predict benefit in human beings or in the person being studied.
And that brings us to the first story, which, if it's okay with you, Pat, I would love it if you would, and I can advance to your slides, if you would tell us the story of dystrophin as a surrogate endpoint. And let me go forward on that. Here we go.
Go to the next slide, Terry. So as Terry described, Duchenne muscular dystrophy occurs 1 in 5,000 births. It is certainly a gene that is on the X chromosome, which means it primarily occurs in males, although we can have X-linked or we can have females that express symptoms. 1 in 5,000 children, about 20,000 people in the United States. This is one of the most common diagnoses in early childhood. The disease, people call it 100% fatal, but lifespan has increased from teens when my sons were here to about the 3rd decade of life. What this disease is these individuals, based on the genetics of one of the largest genes in the human genome, and the gene is 79 exons or 2.4 million base pairs. So we have more than 1,000 mutations across this gene that results in the absence of a protein called dystrophin. So this dystrophin is, as you might imagine, a very big protein, one of the largest in the human bodies, next to titin. So without dystrophin, what happens is muscle, and think about muscle, smooth, skeletal, cardiac, really doesn't survive. So what we see symptomatically in these young boys is that their mean age of diagnosis is 4 or 5 years old. By the time of the diagnosis, it's estimated they've lost about 20% of muscle. So we see in these little children big calf muscles that are called pseudohypertrophic. They can never jump or run in the ways that we think about running or jumping. They don't like to do steps. They don't ride bikes. And by the time they're around 10 or 11 years old, they stop walking. By the time they're 16, they can't lift their hand to their mouth, usually need non-invasive ventilation throughout their adulthood, and die usually because of heart failure. So we now have a gene therapy approval, and we have 4 antisense oligonucleotides that are approved. The antisense drugs are really targeted to people with specific deletions in their gene that, in skipping a single exon, would enable the cell machinery to read that protein. It would be truncated, smaller, but it would be expressed. So the measurement of that expression, so when we measure in skeletal muscle by taking a biopsy and doing a Western blot and then doing all the analysis, the change from baseline, which is 0 dystrophin, to the change after an antisense oligonucleotide is a surrogate marker for benefit. So that is reasonably likely to predict benefit. So what we see in these young boys is when we see expression, then they're usually pushing out those timelines. The sort of the challenge of this story is the challenge, I think, of many rare diseases. This is a slowly progressing disease. So when you do a clinical trial of 12 months, you may not see anything at all. So the approvals have been on the surrogate of expression. The same with our gene therapy, but now because we're using the AAV virus to deliver this gene, the carrying capacity of the AAV is very small. So what we can fit into that carrying capacity is about 30% the size of the big dystrophin. So it's called a microdystrophin. So that delivered 2E14 virus with a promoter into these kids. And again, we're looking at dystrophin, but this time a microdystrophin that we're saying is reasonably likely. In the course of 12 months, if you've seen any of the data and some of the drama around this, the children didn't functionally do better. It is still considered a surrogate. Both Center for Drugs and Center for Biologics still accept it as a surrogate. And to Matt's point about a painful qualification process, it is not qualified under FDA rules. It is expected or assumed to be a surrogate endpoint for Duchenne muscular dystrophy.
So it's assumed to be a surrogate endpoint because it's just a logical mechanistic connection with the cause of the disorder, even though it doesn't actually completely connect to a meaningful change in the family, in the child.
Well, yeah, that's right. So in the studies, both in antisense oligonucleotides and in gene therapy, the primary outcome, which is the outcome that sponsors choose to say that outcome is going to demonstrate benefit, they failed their primary outcome and their secondary outcomes, which are other outcome measures that you're saying are also reasonably likely to predict benefit. What the argument, I think, has been made and the success of getting those approvals is that because of a slowly progressing disease, we look over time. So this is where the phase 4 study comes on, where we're looking and tracking these children over time. So what we do see in both the antisense world, as well as now limited in gene therapy, people who have received those products are walking longer than the natural history. And this gets into some of the report that we just did, is for the FDA to accept alternative and complementary data. That was ACD, right? We had to make up acronyms when we did this report. It was confusing. But basically, natural history data, and this is for all of us collecting data, it has to be rigorous natural history data. It has to be done well so that the FDA thinks that that makes sense and it is true and it is contemporary. So looking at our natural history data along with the outcomes and then looking far out. So we had two ways to prove this to FDA. One is the phase 4 data and looking beyond that 12-month study. And the other is our natural history data, which demonstrates this is a change from that natural history that was contemporary.
Okay, I want to skip around and skip to Matthew and let him start his story, and then Celia after that. And then I'm going to ask a million questions, if that's okay. So can you talk about heparan sulfate and the whole story about that?
Certainly. So the MPS disorders are a group of 12 lysosomal storage diseases, similar to Tay-Sachs in some respects, in that they're inborn errors of metabolism affecting normal lysosomal function. There are 12 of them. They all store a complex carbohydrate called glycosaminoglycan, and we call those GAGs. They come in 3 flavors, so to speak: dermatan sulfate, heparan sulfate, and chondroitin sulfate. And every disorder may store a slightly different version, but we know they store one of those 3 compounds. 7 of the disorders all have neuropathology that is fatal, and they all store heparan sulfate. So we know heparan sulfate causes fatal neurodegeneration, and not just in humans. It does so in dogs and cats and emus and cattle and swine. So it is a conserved cause of neurodegeneration that we know about. Now, in the research field, we all understand that if you lower heparan sulfate, you're getting somewhere with these diseases. But getting that information to an FDA review group in a way that they get it has been difficult. There was guidance that was developed by the FDA exactly about these specific diseases in 2018. It's a great guidance. We didn't feel that it was being used appropriately. And a group of us in the advocacy community, with the leadership of some stakeholders, started to get together to see how we could make some movement. And Pat, you were part of that process, as well as others that are in this room today. We did get a meeting with the FDA. We still felt we weren't being heard quite the way we needed to be. And we fell upon a mechanism called the Reagan-Udall Foundation. Many entities at the federal level have foundations that have been mandated by Congress. The Foundation for the NIH, the CDC has a foundation. The Reagan-Udall Foundation is the foundation that serves the goals of the FDA. And it is an ideal environment to tackle these sorts of thorny issues that may have broad relevance throughout drug development. So they convened a workshop in February, and it was sponsored by 4 industry partners. There were 4 or 5 academics there, and as many advocacy organizations. And we brought to bear data on these 7 different disorders, different kinds of therapy associated with AAV vectors, lentiviral vectors, transplant, enzyme, ERT directed at the blood-brain barrier, all of these things to demonstrate that heparan sulfate being reduced in the cerebrospinal fluid and in the central nervous system correlated with clinical outcome. And I think that was a watershed moment. We had 1,200 individuals attending in person and virtually. 1/10 of those were government, and we assume most of those were FDA. And this was last February. The Reagan-Udall Foundation does a great job of putting that stuff on their website. You can see the content, the webinars live, and a consensus paper they've put out, and then we've gotten one in peer review. Since that date, there have been 4 indications that have been given the green light to use this biomarker and the accelerated approval pathway, which allows drug approval based on a biomarker reasonably likely to predict clinical benefit. They've been given the green light to use heparan sulfate in the CSF as their application moves forward. 2 of those were for a gene therapy, 2 were for an ERT. This is no coincidence. It's been a watershed. And we hope that the workshop example that the Reagan-Udall Foundation has provided will be something that the FDA can pick up with more regular cadence to bring together groups of researchers and stakeholders in a pre-competitive environment. And I know the Rare Hub will be hearing a lot from people on October 16th when they will convene a listening session, I think, on this topic. And I'm also hopeful that the new guidance committee on genetics and metabolism that's been convened by the FDA will also be able to step into some of these questions that really need a thorny issue that need a consensus provided. Thank you.
Yeah. So there's something I don't understand. So dystrophin was accepted because it has a mechanistic connection to the disorder. What's the difference between that theoretically and heparan sulfate, and why was it such a long, it was 2 decades, right? Why was it such a long road to get, that you had to do the Reagan-Udall meeting in order to get through to the reviewers at the FDA?
That's the $64,000 question. We're not sure. I don't know whether it's because our disorders are as 1/10 less frequent. So we're an order of magnitude less frequent in the population than Duchenne for any one of our given disorders. And every program that moves forward in the FDA, it's siloed. They can't talk about that program. So there's not sharing, and it's not malicious. It's just that they have to be confidential about the evaluation of industry programs as they move forward. So I don't think it's been purposeful. It's been a lot of the program reviewers, they're under-resourced. They may not have as much experience as they need. There may have been leadership issues. There may have been a vision or understanding about these things that just didn't help them advance the way they should have.
So I can speak for Duchenne. So PPMD has been around 30 years. We've been involved in or advocating for, like all of you, our disease for a very long time. As we saw the antisense oligonucleotides in development, because it made sense that if you could skip a single exon for certain mutations, if they were amenable to that, and get dystrophin expressed, at least to the patient community, it made perfect sense that that would be reasonably likely to predict clinical benefit. So we went and we met with the FDA on several occasions at the Center for Drugs when Billy Dunn was there. And when we first met with him, he disagreed that that would be a reasonably likely. He said you can't connect those dots. So as this evolved, he said to us, why don't you do preference studies and see if those patients that believe the same thing that you do, right? If you do a rigorous preference study to say what is the priority, then maybe that would be interesting to me. So we did a preference study, and throughout that preference study, we worked with RTI, or the Research Triangle Institute. So it was a very rigorous preference study. So we were the first to do it. And we demonstrated in that preference study that the patients and families' priority was to get a dystrophin protein expressed some way, somehow. So following that, we went back to him with the results of our preference study and said the patient community believes that dystrophin expression is reasonably likely. So we then asked him to write draft guidance, sort of, which is, in our view, sort of a playbook about this disease, so that it would include that. Dr. Dunn said to us, we're not going to write disease-specific guidances. And he turned to me and said, why don't you write a guidance? And I thought, okay. And to be fair, I didn't know anything about what I was saying yes to, but that doesn't typically stop me. So we organized a group, a steering committee, and we wrote the first draft guidance on Duchenne muscular dystrophy, submitted it within 9 months to the FDA. The FDA then turned around and wrote, and this was in 16 and 18, they drafted guidance on Duchenne. So then, more recently, we've updated it. But through those pieces and those interactions with FDA, we were able to demonstrate that we believed, our community believed, and convince them to believe that dystrophin expression was reasonably likely. So I do think it was the pressure of the advocacy community and the activities around in a very rigorous way, presenting data to the FDA.
Cool. I'm gonna switch to Celia in a sec, but oh, I have to go backwards. But while I'm getting my act together here, can you actually explain what a preference study is?
Oh, sure. A preference study is really benefit risk. It is really looking at your community and see how they weigh benefit and risk. How much, and there's a number of different methodologies around collecting that data, and you can use any of those preference. But what you're asking the patient community is how much risk will you take for how much benefit? And these studies, I think, are very important to the FDA so that you can demonstrate to your community. And so it's widespread. You're getting all ages and stages of this disease. If it's a pediatric disease, obviously you're getting the family or the parents. And it is done with much like a clinical trial would be, in terms of we had, I think, 600 people that responded to that benefit risk. And we did it in a methodology that would say, a risk of death is 10%. Would you, if you're expressing dystrophin, would you carry on that? Would you accept a risk of 10% or 20%? So we went down this list of questions so that we understood very clearly what's the preference and what risk are you willing to take to get that dystrophin expressed.
Cool. Very interesting. And I can see how that dovetails right into where I'm going to go with Dr. Goeldner, because her story is a bit different, trying to understand what could be a biomarker for neurodevelopmental disorders, especially 15q. Could you talk a little bit about that?
Yes, of course. So I think Terry invited me today. I'm the outsider. I don't have a big success story of getting surrogate biomarkers approved by the FDA, but we're actually here, I think, with Terry Jo's advocacy in neurodevelopmental disorders and rare disorders in particular, to see how much we can learn and how much we can transfer, and kind of landscape of what were the different steps, where are we today, and how can we move the field forward. So I'd like to talk about 15q chromosome disorders. So I brought a slide just to help explain. So that
Should I go back or leave it here?
No, that's
Leave it here. Okay.
So there's 3 different rare neurogenetic disorders that are caused by genetic alterations in this 15q chromosomal region. There's deletions that cause Angelman and Prader-Willi, and duplications that cause a 15q syndrome. And so there's a set of multiple genes involved, and there's complex imprinting genetic mechanisms there that mean that depending on if the origin of the mutation is from the maternal side or the paternal side, the disorders and the implications of the different genes differ. And on that chromosome, there's a set of 3 genes that code for subunits of a GABA receptor. So GABA is the main inhibitory neurotransmitter in the brain. And there's 3 of those that can co-assemble to form a functional receptor. So it's depicted there. So if you put 5 of those subunits together, you get a receptor that functions and controls inhibition in the cortex and other key brain regions. And here we have a lot of, my biomarker colleague has done a lot of work to explore potential biomarker signatures of that GABA dysfunction in the different disorders. And there's interesting signals. So DUP15q syndrome, which is depicted on the top, has, if you compare their baseline EEG signatures to typically developing children, you have a very prominent peak that you see on the right in frequency called the beta oscillations. And interestingly, in the reverse disorder, if I may call it like that, Angelman syndrome, where when those genes are deleted, you have the opposite phenotypic signature. It's less visible by the naked eye because deletions have, it's the cortical signature is less visible here on the beta band, but you see in that same frequency, so between that 16 and 32, the blue line goes below 0, and that's a decrease in those beta oscillations. And we know from hundreds of years, in particular at Roche, on the GABA-A receptor, because Roche developed benzodiazepines hundreds of years ago, that so we know also that pharmacologically, if you give agents that modulate the GABA receptor, you get exactly those signatures in both directions. So this really indicated to us that these specific metrics of EEG may be promising biomarkers to from both the disease biology aspects, but also for biomarkers to understand the pharmacodynamic and treatment responses of agents that would aim to correct symptoms that are caused by these alterations in the GABA genes in the disorders. And so we had, unfortunately, one of the big phase 2 trials that we were running, which was testing this hypothesis, was not run until the end, but we have on the few patients where we collected data. I'm not showing the data here because it's a bit preliminary, but on the 5 patients that received the GABA agent in children with Dup15q syndrome, we could actually show that indeed that biomarker is sensitive to treatment response and could be used for future drug development. But the story and the road to talking about surrogate endpoints is quite far away. So there's a big difference in a lot of the rare neurogenetic disorders to the 2 beautiful stories that we heard here. And that is in the direct, I would say, monogenic relationship between the cause of the disease and what you're measuring, and also how you're measuring it. So EEG used here, it's a proxy for what happens in the brain. We don't do brain biopsies. You can look at proteins that are floating in the CSF, but you can't probe the brain like you can probe muscles to look at dystrophin, for example. So the links and the claims that we can make are, I would say, less direct. In these disorders, first of all, there are many genes involved, and a surrogate endpoint, a biomarker to be qualified as a surrogate endpoint, it's always for one drug, right? For one mechanism. So even in Duchenne, there's different surrogate endpoints, but they're for different mechanisms of exon skipping or replacement. And so in this case here, for each NDD, that means that we need for each mechanism, for each disorder, to find the exact metric or the exact biomarker endpoint. It's not EEG. EEG is not going to work. It's just a tool to look at the activity, cortical activities, which parameter and how does that link back to the disease biology. And so there's still a long way ahead, but these 3 disorders here are, I think, really a unique situation where we can gather evidence from one to support the other, because I think that's also part of that lengthy process is you need a lot of evidence to make those causal links and make the claims that in the future, would somebody be developing GABA agents for one of these 3 disorders, that just changing the EEG, I don't develop drugs to treat EEGs. We try to do that to treat children and syndromes. So that's one part of the story. And then the other end is what is clinical benefit? And that's the outcome measure section, right, of the discussion. That is also, I think, a big challenge still, getting everybody aligned, understanding in rare neurodevelopmental disorders what is actually clinically meaningful benefit and how do you measure it. We need to really work together, and that's partnerships between patient advocates, industry. We need consortia and the work that Terry Jo does is fundamental in those aspects. Here, I think breakthroughs happen when everybody's saying the same thing and everybody's aligned. has the same goal and has an idea of how to achieve it. So that's food for thought and work for the next 10 years.
Well, one thing I want to highlight. So when I first saw this data, I like jumped on the table and yelled Eureka, because what you left out is, even though it's an EEG and you're not treating an EEG, it was actually connected to the developmental test. It was correlated to the developmental test. Both the Angelman tracings and the DUP15 were correlated to development. And so we might not really care what our kids' EEGs look like, but we do care about their development. And the fact that the EEGs directly correlated to that, to me, was a miracle.
So they correlate in before the drug treatment. So what we know that in Dupi-Q syndrome, the greater that EEG abnormality is, the more severe some of the developmental outcomes are. And what is the key to unlocking all of this is there a correlation of change? So do you see clinical benefits when you see changes on your EEG? That is what remains to be tested. That has not been demonstrated yet.
No, but on the other hand, if you see the EEG normalizing, how different is that from seeing the heparan sulfate normalize, even though it's a slowly developing disorder and it might take a while to see benefit? How different is that from seeing the dystrophin normalize, even though you might not be able to see benefit right away? I think it's pretty, I'm not.
No, I mean, it's that's why we're here to really understand, how similar or different it is and the path to getting to surrogacy. But I think the contribution to the pathophysiology of that cluster of GABA genes, which is what we think that biomarker captures the most directly, remains really to be demonstrated, and that's why we also need better animal models and more preclinical data. It's really building the evidence, because in these disorders there's also another key gene that is thought to be causal, particular in Angelman syndrome, when it's missing or mutated, the UB3A gene. So we don't know how really the contribution to the disease biology, and that puts us in a situation where it's less amenable to predict how changes on the biomarker would relate to clinical outcomes. But it's just really pieces of the puzzle that are still missing. And potentially these drugs may have, a future really used also as combinations. If you have multiple genes that are dysfunctional, you may have add-on clinical benefit, may not be sufficient on its own. Those are questions we don't know. But it would still be possible to demonstrate surrogate efficacy. You need to run the trials and test the hypotheses.
So I would like to ask all three of you, and any of you can chime in here. What advice do you have for the people in the room if they are with disorders that are just starting on this road and don't have, how many of you, can you raise your hand if your disorder has a clear-cut biomarker? We got a couple. All right. Can you raise your hand if you've been desperately seeking biomarkers? Okay. So I guess what I would like to ask all three of you is what tools do you think they need? What advice would you give them on developing biomarkers in the near future? Do they need to collect more EEGs? Do they need to collect more blood? Do they need to collect more natural history? What do they need to do?
I think the first thing is don't let the academics silo data. Because if it's pieces and parts here and there and you have no access to it, you will never find yourself a biomarker. I the academics love to silo data, and sometimes it's not them, it's their institution. I think that the lack or the and NIH grants for a very long time didn't require sharing data, and now they're they encourage the sharing of data. This is patient data. It is not anyone else's data but the patient's, and it needs to be shared so that you can look at all those markers, whether it's imaging, whether it's tissue, whether it's blood or urine or whatever dynamic, whatever you're looking at in terms of trying to understand holistically what is different about this patient, what is the natural history of the change in the proteins in the blood or measuring the urine. You have to get as much data so you can see what bubbles to the top, because you go in blind unless you have a protein like dystrophin, right, or heparan sulfate. But if it's not a single gene disorder, you really have to look holistically at what is changing in these patients in all those parameters to see what bubbles up. And the siloing of data will prevent it.
I think in addition, working from the biology and the physiology of the disorder can be very helpful. I feel like sort of the accomplished older sibling lecturing the younger sibling. The MPS disorders, we've been so privileged because there are multiple different syndromes. We've had animal models forever. Find out what is common about your disorders. What is the common biological pathway? Is it intracellular signaling? What other disorders may have common cascades of disease? Seeing whether you can pool resources, getting that comparative pathobiology understood may point out both a biomarker, and it may help FDA accept the validity of such a biomarker. So in addition to working with the researchers, understanding how to keep data available, understanding the very basic biology, and finding allies in different disorders, in different researchers, et cetera, I think is always a good idea.
Dr. Goeldner?
Am I allowed to make a comment rather than answer the question?
Either one.
Because we've been talking now a lot about surrogate biomarkers, so surrogate for clinical benefits. But I want to make a point that's really important as a drug developer. There's many other kinds of biomarkers. So finding the one that will predict, clinical benefit is maybe the holy grail. And I understand that in rare diseases, of course, time is ticking. So you want to do everything to get those approvals faster. But biomarkers are crucial in all the steps of drug development. So without a biomarker, it's unlikely that you can bring a drug that's been tested only in animals and bring it in the clinic. In neurodevelopmental disorders, without a biomarker, you have no chance to, show brain penetration of your drug. You need to show that you've engaged your targets. You need to understand how much you're modulating it. Is it safe? Is it, the dose selection process, the PK/PD? So finding biomarkers that you already have characterized in your animals that can translate, that you can also measure in humans, is the first step before even thinking of designing a proper clinical study to demonstrate efficacy. You first need to show that your candidate drug does what it's supposed to do. Does it even get to the brain? Does it hit its target? Is that mechanism of action what you thought it was in the dish? So biomarkers are absolutely crucial, and it may never make it to the surrogate endpoint qualification level or validation, but they're all along the drug development chain. They're absolutely essential, in particular in diseases where you don't have great outcome measures, or where it takes too long in neurodegeneration or neurodevelopmental, where in the framework of a clinical trial, it's going to be difficult to capture change. Having these objective measures in parallel is really key to drug development in general.
I also think you need to check with your patient population and see what matters to them. What do they want to see? Because as you're looking at the biology and trying to see a biomarker, you really need to know if what your target is trying to do is important to them, because if they don't care, then I'm not sure you're going to be able to design a trial that's going to be suitable or recruitable, for that matter, to achieve a certain outcome. So you always have to start at the end. What, for instance, in a seizure disorder, decreasing the seizures would be pretty terrific if you had a biomarker and the patients saw fewer seizures in a given day. So I think all of this comes together when you look at that patient population, from the biology and the targets and the biomarkers and the data sharing and what's important to patients.
I think it's also important to remember, and we haven't discussed this part of it, getting an accelerated approval based on a biomarker is the first step in an FDA approval. That will grant you what's called conditional approval. That will allow you to market your drug, which in many of our rare disease indications with slowly progressive pathology is the only viable approach to keep a company alive. But then you have to go through a confirmatory trial, and there you have to reach the clinical endpoints that really relate to the feels, functions, and survives criteria. And there are diseases that are in process. The FDA has a whole site that describes that. So just want to make sure that we put this discussion in the context of what will get you to a full approval and what will get you to a marketing approval, so to speak, within the FDA.
Did you want to say anything else about that, Celia? About the accelerated? We don't have to. I can go to questions. It's up to you.
No, I was having a conversation with Terry Jo before the panel, just thinking broadly also, accelerated approvals also may have downsides. So depending on what you're trying to get approved are drugs with good risk benefits and that do something that's meaningful for patients, right? And there may be trends, right, for companies, because their survival depends on that, to push conditional approvals, because you can start giving the drug, but you do have, as was just said, you do have to demonstrate at some point the true risk benefit of your drug. And there are some fields where it may be a problem if the links are not so strong and the community is not convinced that actually this surrogate biomarker will be predicting something meaningful and clinical benefit for the patients, then other companies working on the same disorders with similar mechanism of actions are also going for similar conditional approvals, and you have drugs on the market that have not shown benefit yet, preventing the other drugs in the pipelines and some early companies to actually bring maybe better drugs to the market. So it's a balance, and it really depends on the data and how scientifically, valid your package is. If you're convinced, go for it. But if
I thought this might bring up a nice discussion.
I mean, I understand that it's you want to get drugs that work approved fast, but we also have to make sure we get the right drugs approved for the patients.
Thank you for that. So this is a perception that has been voiced by the FDA. They worry that accelerated approval for a rare indication will close out others from getting into the market. I think it is exactly the opposite, because you will be able to trial a drug that may do better with that particular biomarker in a head-to-head match with the conditionally approved drug. And it's also an expectation, because of the dire need for indications that are going to be granted accelerated approval. They have to have severe unmet need. There are going to be programs that fail. It's a gamble, and it is accepted, and it is part of the program that some will fail. I believe that many in the monogenic metabolic disease space will fail because a better drug has come on the market and superseded them, and they will be dropped by their programs because something else is better, and it was achieved that status because of accelerated approval. So I think it'll actually drive innovation.
Pat, we
And
only have
that's a
1
good thing.
minute because we got to do
Oh, so I think that's exactly right. We are poster children for having accelerated approval in 4 antisense oligonucleotides with companies who have not completed their phase 3, or if they have, haven't released the data. Either way. But what that has done is innovate in our space with 6 new companies working on a better delivery, better targeted antisense oligonucleotide, and hopefully with better results. So I agree with you. I don't know that the FDA would pull them. I know they tightened their grip a little bit because now they won't, at least in our space, they won't grant an accelerated approval unless the phase 3 is fully recruited, which I think makes perfect sense.
Okay, let's go to questions. And I think, Pat, you're going to let her have your microphone, and then we're going to pass ours along if we need to answer.
So I'm going to start off with one question that we got virtually, and then I know a lot of our in-person folks have had some trouble logging into the app to check in for this event, so we will hopefully have time for 1 or 2 questions from the audience as well. The first question I have is there a biomarker the FDA prefers, or would they accept any so long as they are documented thoroughly?
Any 3 of you want to take that one?
Yeah, we need some
She
is that
said-
additional types. Could you repeat the question?
I can repeat it.
The
I cannot
beginning.
rephrase it because I do not know what it means. Is there a biomarker the FDA prefers, or would they accept any so long as they are documented thoroughly?
Like, does the FDA prefer EEG, or do they prefer a blood biomarker or whatever?
I don't think the FDA has a preference to the biomarker. I think what they want to see that is based on the biology and tracks to potential benefit. So I don't think there's like they're hung up on EEG or dystrophin or whatever. I think they want to connect the dots.
Do we have another question?
Hi. How does the FDA or have you seen luck in industry with using biomarkers that are not necessarily part of the disease or the phenotype, but that they are clearly present with patients that have the disease? As a, for instance, if I was to reduce, I'll just use ours. If I reduced lactate in the brain and spectroscopy, that's actually just a symptom of the disease. The mitochondria are spitting out waste and it collects as lactate in the brain. But that's not part of the disease. It's not part of the function of how it occurs. So is that the surrogate endpoint that you're talking about?
So when we use the term surrogate, it's a frustration to us in the sort of simple metabolic disease, because our biomarkers are what Emil Kakis calls primary disease activity biomarkers. They are as closely linked to the disease as you get. The surrogate term is really a reference to a surrogate for a clinically validated endpoint. That being said, I also know the FDA likes to see layers of biomarker data. So recently, Denali, who's developing drugs in our space, they have heparan sulfate in the CSF that they measure, and it comes down. They also find that neurofilament light chain, which is associated with neuron loss, also comes down. So that's a much more downstream biomarker. It's not integral to the storage disease, but it's integral to the neuronal loss. So more downstream, more data is better, right? I do think the ones that have proximal connections to the disease are valuable, but layering multiple biomarkers that support each other, it's only going to make your case stronger, I think.
Do we have more questions?
We have time for 1 more if anyone has a question. All right.
Can you talk a little bit about, I see a lot of things being advertised as the gene expression being used as a biomarker, like our children's disorder, it's haploinsufficiency in this gene. So we see, oh, we have this assay. If we make the gene expression go up, that's probably going to make things work. Can you talk about, is that real? I mean, just discuss a little bit about that, please.
Celia, you want to take that one?
I mean, it's it's a big question. I mean, haploinsufficiency, I would say in principle, this is what some of these disorders are about, and where you would expect kind of linearly that if you restore the gene function, then you restore the RNA, you restore the protein functional, and you've counteracted the disease. But I think that's very hypothetical and maybe simplistic, and the only way to get there is to have supportive evidence and data. So that needs to be generated. It may be different depending on the gene, depending on what it codes for, if there's been compensations. I mean, depends on the organ. I think it's very difficult to have a one fit for all answer. But I mean, I'm a scientist, but I think the answer is in data, demonstrating, providing evidence, different layers of evidence. It can be direct, indirect in different species.